Thank you, host, for the introduction. Hello, everyone. Good afternoon. My name is Xiaopan from GenScript ProBio. I'm the director of Platform R Research and Development Department in GenScript ProBio. Today, I'll be giving a talk about scalable AAV manufacturing strategies. The talk will fall into some categories. I'll briefly give an introduction to our Plasmid platform as a raw material of AAV manufacturing. I'll talk about the proprietary AAV packaging cell line, or PowerS293 hack-based cell line. And I'll talk about the scalable AAV manufacturing process and how we develop that. So to start with, I'll briefly introduce GenScript ProBio. So this is the history and milestone of GenScript. A brief summary about the history. We were founded in 2002 in New Jersey. We were listed in Hong Kong in 2015, and since then we grew rapidly with the iconic establishment of Legend CAR-T product in 2017, and the ProBio CDMO company in 2020. So last year we formally established our ProBio as an independent company, and this year, up to today, we have 8 approved IND in gene and cell therapy service line just in this year. So this is our strategic business positioning. As GenScript Group, we are focusing on 4 big sectors: the life science service and products, the traditional CRO service. We have the industrial synthetic biology products, and we have Legend offering cell therapy drugs. And we, as GenScript ProBio, we're focusing on biologic CDMO. We want to bring innovation through collaboration. And here we have 2 main arms. One is our gene and cell therapy service products. The other is the protein and antibody drug development and manufacturing. So this is our Okay, we've highlighted some of the major footprints in the gene and cell therapy service line. In 2018, we assisted Legend Bio in obtaining the first CAR-T IND approval from the Chinese NMPA. And followed by that, we established our CDMO segment and ProBio brands in the following years. In 2021, we formally established ProBio as an independent company, as I just said. And we've got the financing from Hillhouse Capital. And this year, we established our subsidiary also in Korea, which is a major milestone for us as well. So in the past several years, we've gotten acknowledgement from the customers of in total 18 IND approvals from China, US, EU, Korea. And we provided plasmid and lentiviral vectors for over 160 big pharma and leading biotech. And here we also included some. It also includes 5 leading vaccine companies in China, which are listed here. Some of them they have the second IND clearance with us, and the Abujan being the fastest one is going into the clinical phase 3, and it's currently undergoing clearance in Ministry of Health. And in total, we have about 150 integrated CMC projects as a whole. So in the next section, I'll be talking about our ProBio cell and gene therapy CDMO business. So in total, we have 4 service lines in our GM cell therapy section, including the lentiviral vector development and adeno-associated viral vector development. We also have a plasmid to support the viral vector development, CMC. And also, we've recently launched our mRNA platform. And today, let's just focus on the AAV service. And here I'll be talking about the associated plasmid platform. And for AAV, we have also a platform, quite a consistent platform for different serotype AAVs. So this is we have team. This is our leadership team. We have a team of expertise to provide our customers with process experiences and scientific knowledge. And our CEO Brian has more than 20 years experience in biologics development, the Amgen, Samsung. We have our operation head in GenScript ProBio, our QA, Vice President of QA, and project management and regulatory affairs, and followed by some of our key members in regional business development. Our main plasmid and viral vector facilities are located in Zhenjiang, which city is 1 hour drive distance from our GenScript headquarters in Nanjing. And our facility, we have our facility construction plan to support consistently to support our customers from the early research to clinical phase and to the commercial. Currently, we have our plasmid and viral vector GMP facilities in this year, and I mean next year. We are further expanding our plasmid and viral vector facility in both China and US. So as I mentioned before, we are the facilities are in Zhenjiang, about 1 hour drive from our headquarters in Nanjing. The plasmid facility is about 6,400 square meters, and the viral facility about 2,500 square meters, and both of them support from research level all the way up to GMP 200-liter scale culture and fermentation. So with that being said, I want to zoom into today the majority of today's talk, which is the AAV vector platform. So we use triple transient transfection system for AAV production, which is still the most efficient way for AAV manufacturing at this time. There are the 3 plasmids vectors carrying gene of interest, the AAV rapid cap and AAV helper genes. These are transfected into our proprietary HEC293 cell lines, being cultured in suspension culture, followed by the AAV encapsulation, release, harvest, purification, and analysis and quality control. So I will give a brief introduction about the plasmid platform, since it's a critical raw material for the AAV viral vector process. Through the past years, we've accumulated significant amount of experience and expertise in developing robust and plasmid manufacturing processes. A typical manufacturing flow chart is depicted here. Upstream process development, we do comprehensive whole strain selection, focusing on identified key performance indicators. And at downstream process, we use mainly 3 steps purification to guarantee robust impurities removal and the performance during the later process scale-up stage. So specifically, we use gel filtration to remove RNA and protein, affinity chromatography to remove open circle plasmid, and AEX to remove endotoxin. The general recovery rates varies between 20 to 60%, depending on the plasmid itself and project. We have overall in upstream, we have overall high density fermentation is up to 1 gram per liter titer. We show here a table of product release items that we pay attention to. It includes, for instance, the appearance, concentration, some key product quality attributes, for example, sequence integrity, supercoil rate, and the impurities attributes. So it's a table for your reference. A free-to-operate strain is critical for stepping into the drug manufacturing CMC. So here we have a clear source of strains and plasmid. We offer that to our customers for both strains and plasmid. We have a clear traceability and regulatory compliance documents to support our customers in IND and BLA filing stages. So here listed our license strain and the payment structure. The license fee is only required to be paid in the BLA or NDA commercialization stage with certain amounts divided before and after BLA approval. And the document list, including the record of the strains and the development records, are comprehensive and complete. So these can be used to support the filing. And we have a clear history of the plasmid from the de novo synthesis at the regulatory compliant platform, with also associated document list and to support the filing. So for AAV specifically, we want to introduce the ITR, which is the inverted terminal repeats region. It's a typical structure in AAV transfer plasmid. This structure is what makes the AAV plasmid AAV plasmid. So ITR has they are inherently unstable due to their secondary structure, palindromic nature. So at the both ends, and with the high GC content, makes the ITR easy to the recombination easy to happen. So the ITR recombination can cause, for instance, the mutant plasmid, low plasmid purity, or abnormal AAV packaging. And that ITR loss can be detected by, for instance, restriction digestion using enzymes like SMA1, which have the recognition site in the ITR regions. And apart from the transfer plasmid, we also have ready-to-use helper plasmid and a rep and cap plasmid to support our customers. And we are going to put them as on-shelf products and ready to be used by any customers. And the current product shelving is ongoing. We plan to have them ready by Q4 this year. And we will have the AAV helper, AAV2 rep cap, and AAV5 rep cap. They've gone through the patent issue, and that should be free to operate by our customers. So to prevent the ITR loss, selection of AAV plasmid producing strain is very critical. So we pay extra attention during the early plasmid CMC step on the ITR recombination and stability of the plasmid. So here during the plasmid CMC, we focus on the ITR sequence and plasmid loss rate during the passage stability. We also pay attention and balance the process overall performance with plasmid yield and supercoil ratio. So here we've shown the data. We've selected different coli strains and in different culture media to test the yield of the plasmid, and also to test the quality, the supercoil rate of the plasmid. And followed by that, we typically also study the stability test of the plasmid during the passaging study. And here listed, we show an example during the strain development. Internally, we have a scoring system, scoring matrix to focus on some of these key indicators like the supercoil ratio, yield, and the loss rate, as well as sequence integrity to select the best clones for the later manufacturing process. So here, as you can see, through different studies, we've defined through different analysis of the growth rate, yield, and supercoil ratio. It shows that strain number 2 and 6 can increase the yield significantly while sustaining a very low plasmid loss rate. And here are 3 cases we've summarized. There are real world cases on our AAV Plasmid production, and one helper rep cap and the transfer plasmid. In general, we deliver the processes with fermentation around around 500 milligrams per liter, roughly, and overall a recovery about 30%, depending on different projects. And some of them are being through the fermentation scale of 100 liter, and these projects are further taken up to 200 to 300 liter at the current stage. To sum up, the AAV Plasmid service advantages that we have in GenScript ProBio, we have a clear IP and traceable strains to help to support our customers with filing. We have a ready-to-use AAV and also LVV helper plasmids with clear IP, and we have LVV plasmids that has gotten the FDA DMF registration, and we have strong track records from our platform was tested by many different projects and have succeeded in supporting them to the IND phase. Cumulatively, we have 18 integrated IND approved projects. As a CDMO, this also helps us to build strong know-how and continues to evolve our technical and regulatory platforms to provide better services to our customers. So let's go into the AAV viral vector production platform and development platform. We're focusing on the suspension culture system here. Here's a brief AAV suspension process flow chart, same as the plasmid. Some of the critical steps I've listed here, including clarification, the removal of cell debris, the tangential flow filtration to concentrate and remove host cell proteins, and the affinity chromatography to capture the AAV, very efficient capture step, and the ion exchange chromatography for the removal of empty capsid AAV. With our proprietary cell line developed, I want to show in the following slides the advantage of using this cell line. Cell line is very important for overall process development. It's really the starting material of the whole CMC, and the good cell line can eliminate a lot of difficulties for later process development. And to start with, we compared our cell line with the benchmark cell line from a vendor. And overall, you can see that in terms of cell growth, it's very comparable with the benchmark cell line, and it has slightly shorter cell doubling time, meaning that it has higher efficiency for the later AAV production, simply because the cell propagation takes shorter time and cells populates fast. And it results in higher cell density and viability compared to the competitor cell line on the culture day 7. So followed by that, we've done a transfection using the cell line. There are lots of studies we've done using the cell line with all sorts of POEs to test the best transfection condition. Here is a snapshot of the one of the experiments that we've done. With all different conditions, we've tested the cell density after the transfection and the viability. We keep following that, and with the optimized results, the best conditions, actually several best conditions, can result in 3 to the power of 13 virus genome per liter crude titer, compared to the it's much higher compared to the competitor cell line. It's worth note that the competitor cell line, we also selected the best condition. We selected through a different rounds of experiments. So it's really a comparison between the best. So to summarize, it shows that and for this kind of study, we also do that regularly as in our CMC process development stage. So the cell line showed high transfection efficiency and results in very good crude titer. Apart from that, we also challenged our cell line in different AAV serotypes. Here we use GFP as a model gene of interest, and surprisingly good, we saw that for all the tested serotypes, the Power S293 cell line resulted much higher product titer compared to the vendor cell line. So it also showed very stable ability. It's not just for certain types of AAV, but consistently for different serotypes, the performance is better. And overall, it has 30 to 50% of titer increase compared to the benchmark cell line. The other thing to consider for the cell line is the scalability during the process development. For this cell line, we've tested the scale up from 5 liter all the way to 50 liter, and from the cell growth and the viability performance, as well as the titer performance, it can be seen that the cell line shows a brilliant stability during the process scale up. And for the process scale up, I want to mention that it's very critical for virus development, because the whole transfection, transient transfection process is very dynamic. So there's a whole lot of experiments done after behind these results. And typically we'll do mixing modeling with, for instance, mixing time model and mixing studies at different scales. And then we do the mass transfer study, for instance, KLA and heat transfer study at different scales. Also the plasmid and the transfection reagent premix study and the rate of addition study will also comprehensively study them during the scale up. After this, the design space and operating space study of each unit operation in upstream and downstream will pay attention to, and results in precisely designed process lock run. So in the process lock run, it's critical because you don't want your process to be too tight, which results in a lot of deviations during your process scale up. On the other hand, you don't want it to be too flexible, because in that sense, your product performance will be lack of control. So as a result of the process scale up and testing different cell lines, we've shown here the performance results in upstream, in process upstream, downstream, and some quality control, some quality results compared to between our cell line and the competitor cell line. So you can see that the product titer and the downstream performance are very, they are quite consistent across different scales in our platform. And overall, compared to the competitor cell line, it not only it gives us better, higher productivity, but also it results in better product quality. Therefore, in downstream process, you don't need to sacrifice too much recovery to be able to get a pure AAV. So in this table, as usual, I listed some drug substance and drug product QC items for your reference. So here, for both of them, we pay attention to the identification of the viral gene sequence. For instance, we also pay attention to the purity and impurities during either product related or process related, the potency of the AAV itself, the safety issues, and also some physiochemical properties for the release of the products. In the next few slides, I want to briefly go over GenScript ProBio services so that you have a better picture of what kind of service that we can provide. So an introduction to our plasmid and AAV service is from research level sample testing and for the IIT, so to say, the investigator-initiated testing. We have a pro-grade production of the AAV, and for IND enabling CMC studies, we have the IND CMC package ready to offer, and through IND to the early phase clinical sample supply, we have a GM pro grade and clinical GMP grade manufacturing. And for the later clinical and commercial stage, we have dedicated GMP facilities and systems to support the full GMP manufacturing. So in the following two slides, we've listed the brief scope of works in our IND enabling CMC packages for both plasmids and AAV. So for each of them, I'm not going too much into details, but here just briefly highlight the advantages. So the plasmid constructs from a gene synthesis. It has clear history of the plasmid. Yes, we have licensed strains for the customer's peace of mind, and we have cell banks of 3-length viral helpers. And we are also constructing the AAV ready-to-use helper plasmids. And we have validated methodology and comprehensive documentation to support. So specifically, the scope of work in a plasmid CMC study includes, for instance, the vector construction, cell banking or strain banking, process development for transfer plasmid, small-scale production and liquid development, pilot scale production, stability, and the dossier summary for the filing. Similarly, for the AAV CMC study, we have for the CMC IND package, we have the AAV packaging test, and typically our customers can first test the AAV packaging on our platform, and then depending on the results, determine what to do next. There's platform demo run and QC testing, very important to establish the process on our existing platform. There's cell banking establishment, process development, formulation study, AAV verification run, QC test, engineering run, stability test, analytical development, followed by GMP batch and stability study, and finally IND submission. So through this, it typically takes about 8 to 11 months from the plasmid to the GMP release. And the service modules, it's highly customizable, and for the manufacturing scale, it also can be customized depending on the requirements of the AAV quantity. So in the final section, I'll give brief introduction to our quality management system. So in here, so overall, we have a comprehensive quality management system. It's the key to our platform, because not only we need to provide our customers with a robust process and a scalable process, but also we need to have a very comprehensive platform to support the filing and to maintain the GMP compliance and things like that. So we have a well-established quality system compliant with the ICH and GMP system, all the way through the testing, production, facility equipment for material and material control and release, and for personnel organization management. And we have also a comprehensive quality management organization, which include quality assurance and quality control. And we have also a well-established quality system for the FDA and MPA for different territories. Overall, we have 40 plus client audit due diligence. They all met client's approval. And we have a client-focused quality tracker, provides end-to-end quality tracking for every project, basically. And we have phase-appropriate compliance system for different drug development stage, from R&D all the way to the commercial. And for the IND projects, we have many track records to keep challenging and keep improving our compliance system, so that it can evolve and it can be developed consistently. And we have China international clients, for instance, USA, Canada, Korea, and Japan, to help us, to give us all-rounded view towards the filing different territories. So our quality system is certified and demonstrated to support many IND successful filings, which are listed here, and also clinical batches. And currently we have more than 60 ongoing integrated plasmid and viral-vector CMC projects. As I mentioned before, they are also keep us improving on our QMS system. So in the last slide, I want to highlight that we are, as one of the GenScript ProBio members, we are also a company we're dedicated to customer first and to ensure top quality products. Also, we want to offer innovative solutions and flexible business models, and to be able to become your most reliable and sustainable biologics partner. So with that, I want to say thank you to all of you, and I'd like to take questions if you may have any.